I am Dr. Jayakumari, Professor, Department of Pharmacognosy, School of Pharmaceutical Sciences. Now, today I am going to take a topic in the course Pharmacognosy and Phytochemistry, Unit 1. Today I am going to take the topic definition of pharmacognosy, sources of crude drugs, types of crude drugs. Introduction about pharmacognosy. It is a science of biogenic or nature-derived pharmaceuticals and poisons. What are crude drugs? These are used for those natural products such as plants, parts of the plants, extracts of the plants, and exudates of the plants which are not in a pure compounds. The natural products are obtained from different parts of the plant, the entire plant, the whole organism of the plant used, and our whole organism of the plant, animal, or microorganism have been used. Part of the organism, like a leaf or flower of a plant, or an isolated gland, or the secretion of the animal organ or other organ of the animal used. Or sometimes an extract and exudate of the animal or a plant origin also can be used. Sometimes the isolated compounds, they are purified, they can be obtained from either animal or plant, also used as a natural products. The pharmacognosy, actually it was derived from the materia medica. Earlier it was coined in the name of materia medica during the 19th century. Definition for pharmacognosy. Pharmacognosy is the study of medicines derived from natural source. As per the American Society of Pharmacognosy, the definition was given by the study. It is the study of the physical, chemical, biochemical, and biological properties of drugs or drug substances or potential of drugs or drug substances of natural origin, as well as the search for the new drugs from natural sources. The term pharmacognosy was first introduced by the C.A. Settler, a German scientist, in the year 1815 in his book Analytica Pharmacognostica. It is derived. The word is derived from two Greek words, pharmakon, the meaning a drug, gymnoscus, to acquire a knowledge of. So totally, pharmacognosy means a knowledge or science of the drugs. This is the book Analytica, Pharmacognostica Analytica book. It is a research PhD book. During his study, Settler was coined out the term pharmacognostica. What are crude drugs? Crude drugs are the dried, unprepared, or unpurified materials of natural sources used in medicine. Crude drugs are the drugs which are obtained from natural sources like plants, animals, minerals, microbes that are used as such without any modification or without any processing except the natural process, drying and size reduction. These are the major four types of the crude drugs. Natural sources are used for the obtain source for the crude drugs. Plants, animals, minerals, microbes. There are different types of natural sources are obtained. There are major part of the natural source plants. Second is animal. Third minerals. Next microbes. Then marine organisms. Last one enzymes. The sources are contribution. So each and every type of natural source, how they are contributed. The major contribution of the crude drugs is plants 70%, animal 20%, minerals 3%, microbes 1%, whereas the microorganism 5%. The enzymes also contribute both animal source as well as plant source 2%. So now coming to the examples of crude drugs obtained from plant source. The crude drug from the plant source, they are started from the lower plant like algae and fungi to higher plants like flowering and non-flowering plants also. Coming to the animals, there are different types of animals are used for the sources of crude drugs, reptiles, insects, and some of the even aves and other animals also. So mineral sources, the crude drugs also obtained from the mineral source. The mineral source are nothing but they are mainly deposited in the near the coastal area of the sea or in the rock places. It can be the types of natural sources are bentonite, talc, kaolin, chalk, and other natural sources. Crude drugs also obtained from the enzymes. The enzymes may be obtained from the plant source or animal source. The example of enzyme from plant sourse, papain, pectin, whereas animal source pepsin and trypsin. Some of the crude drugs also obtained from microbes like fungus, bacteria, yeast, and lichens. So these are the some of the phytocompounds or natural compound. They are obtained from the microbes. For example, penicillin from fungus, streptomycin antibiotic from bacteria, ergotoxin from the fungus ergot, vitamin B12 from the B2 from the yeast, and coumarins, they are derived from the lichens. Lichens are the symbiotic growth of the both algae and fungi. So these are the some of the examples of crude drugs obtained from natural sources. Here this first column represent the sources of the natural source, whereas second was a best specific example. Vegetable means a plant source. If there are number of drugs, for example, fennel, dill, clove, and senna. Animal source, they are obtained from either animal or from the insects or from aquatic animal or terrestrial animal, honey, beeswax, or clever oil. Mineral sources, talc, chalk, most of the mineral sources are inorganic material. They are obtained from the riverbed or seashore places or coastal areas or from the rock deposits. Microbes, fungus, yeast, and bacteria, enzymes, papain, and pepsin. So there are based upon the natural source, the types of the crude drugs are classified based upon its nature and its existence and occurrence and structure. There are two types of major sources of crude drugs: organized drug, unorganized drug. What are the organized drugs and unorganized drugs? Organized drug, as the name itself, they are organized, they have a regular shape, they are the organs of the plant or animal. They are made up of cells, so they are called cellular drugs. They have a definite structure. The drugs are named as flowers, fruits, buds, like insects, like that one. So organized means they have a cellular character. Unorganized means without any cellular, that is called acellular. Unorganized drugs, as the name itself, they are not obtained from any particular organ of the plant or animal, but they are obtained from either plant or animal by some process of extraction, or these are the natural exudation of the plant or animal. Sometimes they are obtained by simple incision or anything. So after that one, they can be purified if it is necessary. So these are the major comparison between the organized and unorganized drug. Organized drug are the part of the organ of the plant or animal, whereas unorganized drugs are they may be a plant or animal or a mineral origin, but they are not a organ of the plant. They are the secretions or exudations of the plant or animal. They have since they form an organ of the plant or animal, they have a cellular structure internally. It does not have any definite cellular structure since they are secretions of the product, the plant source. Generally, they are identified by the morphological character, but these are identified by the organoleptic character since they are not organ. So we cannot identify any shape or size, surface character, but organoleptic characters, the organs characters which are observed by our sense organs like tongue, eyes, nose, and skin. Examples for organized drugs: digitalis leaf, cinchona bark, ephedra stem, fennel fruits, clove flower. Like that, we can tell many of that one. Even the whole plant also used. Example, vinca rosea, ephedra, like that one. Whereas unorganized drugs, there are different types: animal source, plant source. In animal, example, gelatin and honey. In plant source, example, agar, acacia, tragaga. So they are mostly obtained from plants and animal by simple extraction process or natural secretion process or dried juices, like that they are collected. So this is a flow chart which represents the organized and unorganized drugs, or the crude drugs. There are two major types, organized and unorganized. The organized drugs, again, it can be subdivided into overground, that is above the ground plant and underground plant. Above the ground plant, example, entire herbs, leaves of the plant, flowers, fruits, seeds, bark, and root of the plant. Whereas underground plant, they are below soil. Example, roots, rhizomes, underground rhizomes, modified stem structure, and everything. Coming to the unorganized drugs, as the name itself, they are dried juices of the plant, latex of the plant, extract of the plant organ or animal organ. Maybe their major compounds are presented are the alkaloids, glycoside, tannins, resin. Sometimes the resinous substance, they are called excretion, exudations. Sometimes the oily substance can be separated. Fixer oils and fats, waxes, they are natural fatty secretion, gums and mucilage. So these are the, either they are obtained by simple incision process or simple extraction process, or sometimes some of the unorganized drugs are like waxes and the mucilage as natural exudation of the plant. Coming to the microorganism as the sources of the drug, it also plays a vital role. The Penicillium notatum, it is a fungus. It is used for the production of the penicillin. It is in a broad spectrum antibiotic. Actinobacteria, it is a major bacteria produce streptomycin. It is an aminoglycoside antibiotic. Aminoglycoside antibiotic like gentamicin, amic acid, tofurmycin, these are obtained from the bacteria Streptomyces and Micromonosporus. So again, the crude drugs are for the organized, and it is a difference between the organized and unorganized drug. Organized drug have a cellular structure, whereas unorganized does not have any cellular structure. So it is absent in unorganized drugs. Organized drugs obtained from part of the plant, example, fruit, seeds, flowers, bark and leaf. Unorganized drugs obtained from the plant or animal by extraction, distillation process, incision process, or the expression process. These unorganized drugs can be identified or can be studied by the microscopical method, macroscopical method like transverse section, longitudinal section. They are used by, because they have a cellular character, we can identify with the help of microscope. Whereas unorganized drugs, they are not have a cellular character, so we cannot identify by the microscope. Means they does not form a definite structure or a definite organ or any cellular character. So they can be identified or studied by some organoleptic characters or physical characters like density, viscosity, refractive index, optical rotation, and other criteria. They are solid. Most of the organized drugs are solid in nature, whereas unorganized drugs are, they may be a solid or liquid in nature. For example, fixed oil, volatile oils are, and sometimes they may be a semi-solid like resins, balsams, and wax and other things. Examples for organized drugs: clove, flower, fennel and dill fruits, digitalis leaves, cinchona bark, the whole plant ephedra, like that one. Whereas in unorganized drugs, the liquid substance, example, lemon oil, castor oil, the solid starch and catechu, the semi-solid, example, spermaceti and beeswax, wool fat, lanolin, like that we can tell. So thank you, students. I hope it is very useful for you.